Before we study Mendel's monohybrid and dihybrid experiments. Now let's study about chromosomes, DNA and genes. The human body is made up of millions of cells. Each cell contains nucleus. Nucleus is the controlling center of the cell. The nucleus consists of thread-like structures called as chromatin fibers. These chromatin fibers condensed at the time of cell division or at the time of prophase stage. These chromatin fibers condensed and they become distinct chromosomes. These chromosomes duplicate themselves and form identical copies like itself called as sister chromatids. So at the time of cell division, each chromosomes duplicates themselves and form identical copies like itself is called sister chromatids. Each chromosome is composed of DNA. The short segment of DNA is called as genes. Genes are responsible for the inheritance of hereditary characters. These are responsible for transmission of characters from one generation to another generation. During the cell division, this genetic information is transmitted to the next generation. Let's study the structure of chromosome. Structure of chromosome. These chromosomes are long, thin and thread-like structures. It is consists of double-stranded structure. Two identical strand structure is there. These identical strands are joined at a particular position. This is called centromere. These two strands are called as chromatids. These two chromatids are meet at a particular point, that is called as centromere. Each chromatid is made up of the spirally coiled thin structures. This is called as chromonema. So each chromatid is composed of spirally coiled these thread-like structures. These spirally coiled structures are called as chromonema. The number of bead-like structures are present throughout the length of this chromonema is called as chromomeres. So many bead-like structures are present at the chromonema throughout its length, that is called as chromomeres. This chromosome is composed of DNA, RNA and chromosomal proteins such as histones and non-histones and certain metallic ions. These substances are help to it gives structural support to the chromosomes. This chromosome consists of certain regions. First is the primary constriction. This is the primary constriction. As we studied, this chromosome contains two small strand-like structures, that is called as chromatids. These two chromatids are joined at a particular place, that is called as centromere. This centromere is also called as primary constriction. This is the region in which the spindle fibers are attached to the chromosome. We know the spindle fibers are proteinaceous fibers that is formed at the time of cell division. So the spindle fibers are getting attached to the chromosomes at this primary constriction. This is also called as centromere. Next is secondary constriction. In some chromosomes possess these secondary constrictions. The secondary constriction is formed at any point of the chromosome. These are also known as nuclear zone or nuclear organizer, because this secondary constrictor region produces nucleolus of the nucleus. We know inside the nucleus small spherical structures are there, that is called as nucleolus. So this constrictor region, this is called a secondary constriction. This secondary constriction is also called as nucleolar organizer or nuclear zone. These nuclear organizers are for producing the nucleolus of the nucleus. Next is the telomere. The end of the chromosome is called as telomere. The end of the chromosome is called as telomere. That each extreme end of the chromosome has this polarity. This polarity prevents the chromosome from joining with the adjacent chromosome, and these telomeres also help to maintain the stability of the chromosome. So telomeres are nothing but the extreme end of the chromosome is called as telomeres. The next region is satellite. This knob-like structure is called as satellite. In some chromosome, this knob-like appendages is present at one end of the chromosome. If that appendage present in the chromosome, that is called as SAT chromosomes. Now let's study the types of chromosomes based on the position of centromere. The types of, we know the centromere, it's a meeting place of the two chromatids. Here two chromatids are there. These two chromatids are meet at a particular point, that is called as centromere. That is also called as primary constriction. Based on the position of centromere, the chromosomes are classified into four types. The first is the telocentric. In this telocentric, the centromere is present at the very end of the chromosome. It is a rod-shaped chromosome. This is telocentric. The extreme end, at the extreme end, the centromere is present. Then acrocentric. This acrocentric, the centromere is present near at one end. So here one chromatid arm is very small arm and another one arm is long arm. So this acrocentric, it is the centromere lies near at one end. It has a small arm as well as one long arm is present. The next is submetacentric. So in the submetacentric, the centromere lies near the center. Not at the exactly at the center. That means it is that centromere is present in between the end as well as the center. So this it is called as submetacentric. So it has centromere is present near the center, and here also one small arm and one long arm is there. It is in the shape of letter J or L-shaped chromosome. The next is metacentric. This metacentric is the centromere present exactly at the center. So it is V-shaped. Both the arms are equal in size. And this is a V-shaped chromosome. Next, the types of chromosome based on function. In our body, in human body, there are 23 pairs of chromosomes are there. In this 23 pairs of chromosome, the 22 pairs are called as autosomes, or that somatic chromosomes. The 23rd pair, that is called as allosomes or sex chromosomes. The first 22 pairs of chromosomes, these are called as homologous chromosomes. Why it is called as homologous chromosomes means these chromosomes are arranged in pairs. That means one chromosome we receive from our father, one chromosome we receive from mother. That is why these chromosomes are in pairs. So in human, there are 23 pairs of chromosome, or 46 number of chromosomes are there. Here, the first 22 pairs of chromosomes are called as the autosomes or somatic chromosomes. These 22 pairs of chromosomes are determining the body characters. Then the last pair of chromosome, that is the 23rd pair of chromosome, is called as allosomes or sex chromosomes. In females, that 23rd pair is XX. In females, both the chromosomes are same, XX chromosomes are there. In the case of males, the 23rd pair is in the form of X and Y in all the cells. So in human beings body, totally 46 chromosomes are there. Out of 46 chromosomes, 44 chromosomes or 22 pairs of chromosomes are determining the body characters, or responsible for the body characters, that is called as autosomes. The 23rd pair, the last pair, is responsible for determining the sex of an individual. If it is female, that chromosome is the same chromosome is there, that is X and XX. If it is female, that 23rd pair is X and Y chromosomes are there. Since it is determining the sex of an individual, the 23rd pair is called as the sex chromosomes. Next is karyotyping. The karyotyping is a technique by which the complete set of chromosomes is separated out from the cell, and then they are lined up in an ideogram. It is a graphical representation or diagrammatic representation of chromosomes is called karyotype. In karyotype, we could know the number, size and shape of chromosomes of a particular organisms. This karyotype is performed usually at the metaphase stage. During the metaphase stage, all the chromosomes of the cell is separated out and arranged in the homologous pairs according to their length. It is arranged according to their length like this. Their decreasing length, thickness and position of the centromere and shape, etc. So in today's class, we studied about the Mendel's dihybrid cross and the law of independent assortment. Then we studied the structure of chromosome, the types of classification of chromosomes based on the position of centromere as well as based on its function, then karyotype. Home fun. Students are asked to study the following questions. The answer in brief you should write as contest. Answer in detail you must study, but you no need to write as contest. Answer in brief. First question, what are allosomes? Second question, explain the structure of a chromosome. Third question, under which conditions does the law of independent assortment hold good and why? Long answer, explain with an example the inheritance of dihybrid cross. How is it different from monohybrid cross? Thank you.